Assalamualaikum warahmatullahi wabarakatuh. Let me introduce myself and my team. I'm Betris Fitria Marga, a student of Biology Department, Faculty Mathematics and Natural Science, Universitas Lambung Mangkurat, Banjarbaru, Indonesia. And in this research, in my present today, I'm supported by my lecturer, Dr. Abdul Gafur, MSc, MSc, PhD, as corresponding author. In this opportunity, I would like to present my research about soil nematodes of the Duku orchard in Tabalong Regency, South Kalimantan. The outline of my presentation: the first, introduction; the second, materials and methods; third, results and discussion; and the last, conclusion. Duku plant. The stem of Duku plant has an upward growth direction with a height of up to 50 meters. The bark is broad, rauque, and scaled. The leaves are pinnately compound leaf. The tips of the leaves are tapered on the surface. The color is shiny while at the bottom they are pale. The fruit has an oval orange shape with a diameter of 2.5 to 5 centimeters. The color is grayish yellow and has segmented flesh. In addition to the fruit can be consumed, other parts of the plant are also useful. Nematodes. The name of nematode comes from the Greek, which means yarn. The nematodes are elongate, tube-like, sometimes coil-like, moving like snakes. Nematodes are shaped like tiny worms with a length of 200 to 1000 microns. Generally, there are two types of nematodes. Number 1. Parasite nematodes. Parasite nematodes live inside a plant tissue and they take the food in the tissue by using a stomatostylate and odontostylate. Number 2. Free-living nematodes. Free-living nematodes live outside a plant tissue. Types of free-living nematodes modes can be seen in the following figure number 2. Free-living nematodes, namely bacterial feeders, algae feeders, fungal feeders, predator, and omnivores, play an important role in the destruction of organic matter in the soil. Nematodes are an important indicator of environmental health, where the higher the diversity of nematodes is expected to reduce the dominance of harmful nematodes and increase the role of beneficial nematodes. Background. First, Duku plant is seasonal plant that includes superior fruit plant in various regions, especially in Tabalong Regency. But Duku production on 20 and 16 experienced a reduction from the previous years, which is only 2.79 tons per hectare. Production decline is caused by weather, presence of pests, and disease. The disease can lead to production decline which is caused by insects, fungus, virus, and nematodes. However, there has been no research about the type of nematodes that attack Duku plants, so there was very little information about nematodes existence of Duku plant, especially at South Kalimantan. This research was conducted in January 2019 to February 2020 in three locations. Those are the Nawenhuileran Village, Nawenhuiler Village, and Hayuk, Harway Subdistrict, Tabalong Regency, South Kalimantan. Observation of morphology and morphometry in nematodes was carried out at the Biosystematic Laboratory, Faculty Mathematics and Natural Science, Universitas Lambung Mangkurat, Banjarbaru. Materials. Soil samples. Hot 4% formaldehyde as 1 solution, as 2 solution, alcohol absolute, spiritus, silk gel, paraffin, immersion. Tools. Swavel, plastic bag, labels, pencil, GPS, standard sieve with a size of 35 micron, spray bottle, fill bottle, beaker glass 100 ml, petri dish, filter, tray, object glass, cover glass, pipette, microscope, stereo and binocular, hot plate, Bunsen, and top clamp. Methods. The first, soil sampling. Soil and plant root tissue, samples were taken a depth of 0 to 20 cm because the highest nematode density and population were found at the tip. The taken soil samples are put in plastic bag that has been labeled. Then the soil and root tissue samples were taken to the laboratory to identify in the biosystematic laboratory. Extraction and isolation of nematodes. The method used is the whitehead and hemming method. The soil sample is put on the top of the sieve which layered with tissue paper. The function of the tissue paper is to keep soil so it doesn't fall into the tray. Then they are soaked in half and there is a gap between the bottom of the filter and the bottom of the tray, so that there is a space for the nematodes to escape the bottom of the water. The soaking process is carried out for 2 days. The result of sediment is the tray is filtered with a 45 micron sieve and put into a petri dish. The nematodes are transferred by a microscope stereo and a simple tool of a brush that the tip has been trimmed and put into the end of the bamboo blade. This method is considered easier and cheaper to be applied. Preservation of nematodes. The preservation method used is the sandhorse method, which is hot formaldehyde with 4% rates and room temperature formaldehyde with 4% rates are put into a test tub with a ratio of 1:1 and let it to stand for 2 weeks. After that, the nematodes were put in a petri dish containing S1 solution and placed in a closed container filled with 100% alcohol. Then put in an oven and 400 Celsius for 24 hours. S1 solution was replaced with S2 solution and put in a closed container filled with silica gel after 2 to 3 hours. Then treat it with glycerol so that the nematodes don't dry out. Mounting. The paraffin circle was placed in the center of the slide, then drop glycerin on the center of the paraffin mold while the nematodes are placed on the center of glycerin. The nematode is positioned sideways on the slide for easy observation. The slide is held until the paraffin melts. The slide is cut and covered with a cover glass. Morphology observation and identification. The data analysis was given qualitatively in the form of morphological description of the nematodes and quantitatively in the form of nematodes morphometric measurements. Results and discussion. The nematodes that were identified in the soil sample of Duku plant in the Tabalong village were the genus Eotenchus of the order Mononchida as much as 2 individuals. In the nominhular village were the genus Westinincus of the order Dorilaimida as much as 1 individual, and were the genus Ironus of the order Anophlida each. In her village were the genus Westinincus and Ironus 1 individual each. Not all nematodes can be identified because the nematodes obtained are young or immature nematodes. The nematodes organs have not developed properly. Eotenchus. The genus Eotenchus found were female, while the male were not found. The female Eotenchus were found that have body length and diameter ranging from 1.48 to 1.85 millimeters and 66.6 to 74.51 micron. With smooth cuticula and its lips are offset. The oral cavity or stoma is thickened with a length of 35.48 to 40.93 micron and a weight of 25.71 to 27 micron. The position of the vulva is 49 to 55% from the anterior. The tail extends toward the filiform with the length range from 122.2 to 352.26 micron. The genus Vestinicius found were female, where the male was not found. After the fixation process, it found that the upper posterior body curved into the ventral side. The body length and the diameter ranging from 1.64 to 1.97 micron and 53.25 to 67.90 micron. The cuticular arctic and smooth offset lips have a lip width of about 22.96 to 25.77 micron and a length of 8.56 to 10.96 micron. Brachyolostoma with four-legged onychia cling to odontostylate. The stylate's length range from 20.94 to 26 and 38 micron. The wall of the stoma has small denticle, double breathing ring from the anterior end. Odontoper is shaped like a root. Developing reproductive system. The vagina is sclerotized. The tail is gradually tapered with a length of 145.91 to 151.24 micron. The genus Ironis found was female, while the male was not found. The nematodes of the genus Ironis were found to have a body length of 1.7 millimeters with smooth cuticula, and its lips are offset by 6.96 micron length and 14.71 micron wide. The oral cavity or stoma has a cylinder shape with 7.22 micron length. The stoma extends from the base of the lip to the esophagus. There are teeth located at the anterior tip of the lips. The female reproductive system is self-contained. Two ovaries with the vulva position at 40% from the anterior end. The tail is very long and tapered toward the tip with a length up to 434.2 micron. Conclusion. Based on the result on study can be concluded that nematodes that found on Duku plants soil in Tabalong Regency was Eotonchus from ordo Mononchida, Vestinicius from ordo Dorilamida, and Ironus from ordo Phenoptida. The soil nematodes found in Duku plants in nominhilliard village are genus Eotonchus, in nominhilliard village are genus Vestinicius and Ironus, and in Hayu village are genus Vestinicius and Ironus. This is our references. And thank you very much for your great attention. Wassalamualaikum warahmatullahi wabarakatuh.